I am Dr. Terry Jo Bichell. I am a neuroscientist, and I'm also the mother of a 22-year-old son named Lou with Angelman syndrome. And I'm very excited to be doing Angelman Updates, which are a new venture co-produced by the Angelman Syndrome Foundation and the Foundation for Angelman Syndrome Therapeutics. And we are really excited to be interviewing everybody who's doing the coolest cutting edge research in Angelman syndrome. And so I am really excited to talk to you today and learn about your research. So Dr. Albert Keung, welcome to Angelman Updates. And if you could please just introduce yourself just a little bit.
Oh, sure. So my name is Albert Keung. Let's see, I was born in Hong Kong, but moved at a very young age to a suburb of Boston. Always been really interested in science and did all my schooling in the Bay Area and started my lab here at North Carolina State University in 2016. Yeah, so looking forward to telling you more about the work that we're pushing forward.
So how did you get into the Angelman field? What got your toe wet in the Angelman world?
Yeah, so my graduate work, it probably started, the experimental systems that we work with is probably how we got into the field. So in graduate school, my PhD work, I was studying neural stem cells, and we were moving more towards human stem cell systems to try to capture a lot of kind of the human-specific aspects of biology. And so when I started my lab, we were interested in improving those models, improving these human stem cell derived models of the brain of neurons.
So, but you could have studied one of 500 different neurological disorders. Why Angelman?
Yeah, and I think it's there were a combination of things. So being at North Carolina State, UNC Chapel Hill is our sister institution, just 30 minutes away. And UNC has really strong research in autism, as well as Angelman syndrome, with a few of the pioneers being there. And so when I was reading about what we wanted to work on, that kind of caught my attention. That we had some local experts. So that was definitely one aspect. The other aspect is that a major focus of our lab is studying epigenetics. And this is, of course, a very central problem mechanistically for Angelman syndrome. And so I think all of those things kind of combined together is what led us to Angelman.
So, okay. So you said you started your lab there in 2016. So you've been working on Angelman experiments for 4 or 5 years or less, or how long?
I think so. Our first ever grant funded to do work in this area was I think like towards the tail end of 2016. So I'd say when we first started, we were first just developing organoids. Back then, these kind of micro tissues were quite new at that point, and so we were developing the expertise to be able to work with develop organoids. And then the Simons Foundation funded a small pilot project for us to map UBE3A localization in human organoids. And so we did that, and that's kind of how that was really our first project, just figuring out where and when UBE3A was expressed within different cell types and different regions of these kind of micro tissues that resemble different regions of the human brain.
So you almost said mini brain, but then you stopped yourself. Is that term not in vogue?
I I avoid it because it really isn't a mini brain, and I think it evokes consciousness or ideas of a consciousness or thought, these types of things, when in reality they're very small. They don't have complex networks of neurons interacting with each other. So from an ethical perspective, we're very comfortable working with these systems. Whereas, obviously we would be very cautious working in a system that's more reminiscent of an actual human brain.
So this is more like a pile of wires than it is like a robot.
Yes, yeah. Yeah, I would say like it's a lot of parts, but maybe not put together. Yeah.
So you can sort of see how the parts might interact. So if you zap one wire, which in your case, if with the organoid, would be zapping one of the neurons that forms in the organoid, it might connect to one of the other neurons, but it's not going to connect in the same way that it would make a thought.
That's right. Yeah, I thought, our brains have trillions of connections, probably more. And thoughts are really probably composed of large circuits of many connections, and not just one circuit, but many circuits going on at the same time. In the organoids, I think the most complex we would get is one neuron firing and connecting to another neuron.
So you've been working on these organoids, or you've been getting them going. What have you found out so far?
Yeah, so our one of the big questions that we wanted to ask was, when and where. So the UBE3A protein is a central protein for Angelman syndrome, and there's been a lot of studies in mice that have kind of tracked where UBE3A shows up within a cell. So is it in the center of the cell, the nucleus where the genome is, or is it out in the cell body? And also, when does it turn on and when does it get silenced? All of which are really important for therapeutics as well. And in mice, it seems like there was a critical time period where a lot of these transitions, when the paternal UBE3A was being silenced, when the protein was moving into the nucleus, a lot of these dynamic changes, it seemed like these were all occurring very close to birth. So around postnatal time periods in a mouse. So in a mouse, that's 14 days or so. And clearly human gestation is much longer. It's 9 months. And so one of the things that we wanted to ask and answer was, well, in a human context, when does this protein get paternally silenced? When does it move into the nucleus? Because it could have implications for therapeutics, therapeutic delivery, the timing of that, and also understanding the function of the protein and whether in both development, in actually human development as well. And so what we found, so the organoids are a really good model because they can model prenatal time periods, which is something that is really hard to access. You can't access human systems inside the womb, of course. And also it's hard to access that even outside of the womb, right? We can't experiment on humans, of course. So the organoids are a really nice model because they actually create a lot of the cell types and track the kind of temporal development of a prenatal brain. And so what we found is that it seems like there's a period between the first and second trimester when equivalent to the first and second trimester in these organoids when the UBE3A protein is going to the nucleus and when the paternal UBE3A is being silenced. So that's kind of one of the key findings. We also, we also wanted to just test whether these models were good for looking at drug responses. So for example, indotecan was one of the first chemicals that was shown to reactivate UBE3A. So we added indotecan to the organoids and showed that that could reactivate the paternal UBE3A, and actually that one dose was able to last for quite a while, for several weeks actually, which was similar to what was reported in mice by Mark Silka and Ben Philpott several years ago.
So, okay, so what you're saying is that in these little organoids, they are sort of like a developing fetus in a way. I mean, they're not. I don't want people to get mixed up and think that you're doing fetal research, because you're not. You're doing research on cells in a dish, and stem cells are not fetuses. And I just want people to be really sure about that. So whenever we talk about this, we're talking about cells in a dish that are not connected to any other, organ. And those cells are a way for us to study humans without hurting any humans and without touching any humans. That's so important. So what you found in this is that at around the 3 months gestational phase, more or less, because it can't be exactly the same, we don't know if the cells in the dish, but getting close, that the gene that causes Angelman syndrome moves into the nucleus of the neurons. Is that what you're saying?
Yeah, that's right.
And so what I remember was that whenever neurons became active adult neurons, meaning that they were actually able to fire and act like adult neurons, that was when UBE3A was, the paternal was turned off. So are you saying that at 3 months gestational age equivalency in the dish, that the UBE3A moves into the nucleus, or are you saying that at that age is when the neurons become active and that's when everything happens? So it's only just whenever the neurons become active.
Yeah, so this is one of the, that's a hard question to, it's a very good question. It's just a hard question to answer with the organoids, because I'm pretty sure that the electrical activity of the neurons in an organoid are nowhere near that of what you would see in a developing fetus. But I would say that in our hands, I would guess that UBE3 is moving into the nucleus actually in neurons that are not yet electrically active, potentially, because we actually see many UBE3A moving into the nucleus in some neurons, even at a month or 2 months. It's just that the organoids also still have neural stem cells and transit amplifying cells that are not differentiated into neurons yet. And so these organoids are actually heterogeneous throughout the 1, 2, 3 month periods. They still have stem cells in them. They have neurons of different maturation stages. Okay. But even 1 month in, we can see paternal silencing and UBE3A moving into the nucleus.
Huh. That's so interesting. So then what about, well, now I'm just being nosy, but if you have, you know how I remember when we found out that you can have stem cells making new neurons your entire life. I remember, up until 20 years ago, we thought that whatever neurons you had, you never were going to make any more. And now we know that you can actually make new neurons through your life. So those new neurons that are the stem cells actually that the brain itself continues to make in certain little areas of the brain, are those neurons with the UBE3A in the nucleus and the paternal turned off, or are those neurons already past the phase where the UB3 has already gone into the nucleus and what's going on there?
So so in adults, we have a slightly debated topic in the neural stem cell field, but a sizable proportion of neuroscientists believe that in adult humans, in the hippocampal region and in the subventricular zones, the two parts of the brain, there are kind of in small niches, environments in which there exists stem cell pools that are, can still divide and make new neurons. Because the organoids are they don't form brain structures like a hippocampus or SVZ, we can't necessarily say that we've mimicked those exact cell types. But what I can say is that there are stem cells in the organoids that have similar signatures to, for example, the stem cells in the adult SVZ or the adult hippocampus. And in those stem cells, the paternal UB3 is not silenced. And it doesn't seem like the UB3 is nuclear in those cells either. Which is similar, I think that was also observed in a mouse, in the mouse model of an adult mouse.
So do you have all different genetic forms of Angelman syndrome in your organoids, or you just concentrate on certain forms? I mean, which ones are you working with?
So our initial work for the past, 3 years was working with just neurotypical organoids, so organoids that didn't have any mutations or deletions, and we were just characterizing UBE3A. But but recently we've been doing comparisons of organoids that have UBE3A knocked out, like both copies actually removed. And we actually got the stem cell line from Stormy Chamberlain at University of Connecticut. And they're very nice in sharing their lines. And actually what FAST is funding us to do right now is to actually generate stem cells that have that mimic the genetic changes, different genetic backgrounds of Angelman syndrome. So Angelman syndrome is actually a diverse disorder where you actually, it's not every patient has something unique genetically, and we want to be able to develop models that allow us to understand that diversity, maybe screen for drugs that for each genetic difference. And so what we're doing right now is creating stem cell lines that have class 1 deletions or versus class 2 deletions, uniparental disomy, and potentially use these systems to understand how the other genes that are lost in class 1, class 2 deletions, for example, might also be important, not just UBE3A. Yeah, so that's definitely where we're moving. We want to be able to generate models that can help us understand, potentially develop drugs for many of the Angelman individuals that do not just have a UB3A alone deletion, but they have deletion of other genes or unique mutations and such.
Are you taking cells that don't have the Angelman genetic issues and actually creating the Angelman genetic issues so that you already know all the other genes, or are you taking donations from families and patients and using those cells to make the new samples.
Yeah, so those are definitely two very complementary paths. And so we are doing the first path, although we're also working with cell lines or generated through the second path, what you just mentioned. So actually, Yale, there's a fast run program at Yale that is having donations from Angelman patients and like skin samples or blood samples and converting those into stem cells. Um, and and then so that's very useful because you can then look at the differences between the cell types, the different genetic differences. But one of the challenges is that humans in general are very, or human samples are very diverse. So even the same genetic background, so if you just you could take two class 1 deletion cell lines, for example, and then compare them, there might actually be quite a bit of just inherent natural variability in how they behave. Because there's a lot of other differences in our genes and in our genetic sequence. So, so we also wanted to take a parallel approach to that, which is where we take one cell line and then we're modifying it to have the same genetic changes that appear in Angelman syndrome. So either a class 1 deletion or maybe a mutation in UBE3A or uniparental disomy. And what we hope there then is that because everything else in that cell, in those two cells, the edited cell and the original cell are the same, then when we compare differences, there will be fewer inherent differences, and hopefully the differences we see will be arising largely from the Angelman-related genetic differences.
So just to rephrase that, so what you're doing is taking a line of cells from a human donor, but then what you're doing are creating in those cells each of the different kinds of mutations or variants that cause Angelman syndrome. So you're making maybe a point mutation in UBE3A or a deletion of UBE3 or a class 1 size deletion or a class 2 size deletion, and or the two uniparental disomies, but it's basically all the cells are really from the same original donor.
That's right.
So nothing else is going to change. So it's kind of like identical twins, only one of them has the mutation that causes Angelman syndrome. I actually have a set of identical twins.
Oh.
Not my son with Angelman syndrome. He's another one of my kids, but I do have a set of identical twins. My whole life's a genetics experiment. That's what I always tell people. But so basically you're creating quadruplets, quintuplets, all these different cell lines that are all the same except for those mutations that you make. And that way you can really compare. If you see a difference, that difference is only because of the change that you made. There's no difference in the rest of that cell line or the rest of that every organoid or
That's right.
the
Yeah.
brain on a chip or whatever it's
The
called.
twins analogy is really a good one. Yeah. Yeah. Basically identical twins. So everything's the same genetically except one difference, genetic difference. Yeah.
That's so interesting. And you actually are working with Dr. Jiang, who I just interviewed earlier today, but I wasn't sure if you were working with them. So that's fantastic. So you can actually compare the ones that you have developed, which you have caused those mutations. You can compare those eventually with the same ones that Yang Wei will be developing, which derive from actual donors that have Angelman syndrome. That'll be interesting too.
Yeah, and I think we'll we'll hopefully have multiple sets of iPSCs from Dr. Jung's lab at Yale and edited versions. Some, for example, class 1 deletions are very difficult to generate using molecular tools. And so one of the things that we're hoping to do is actually take one of the class 1 deletion lines from Yale, and then what we can do is rescue, meaning we can reintroduce many of these missing genes. So not just UBE3A, but there's also GABA receptors, which are potentially very important for a lot of the phenotypes and the symptoms in Angelman individuals. So yeah, that's one of the, I think there was a lot of feedback and kind of going back and forth amongst investigators. So it's one of the powers, I think, of what FAST and ASF are doing.
That's so cool. So you can actually put back in that whole sequence. How do you put it back in? Do you borrow it from the paternal side and stick it in? I mean, you have all the little pieces that are in between the genes too. How do you make sure that you get everything that matters in there when you're replacing it?
So it completely recapitulating everything just like it is will be difficult. And the best way to do that would be to start with the full sequence there, and then kind of snip out different parts and then understand what happens when you do that. But there's also, I would also call myself a synthetic biologist, meaning that we engineer cells to understand how they work better. And one of the kind of approaches in synthetic biology is that you can put genes under the control of a chemical or small molecule or even light, and tune how much of a gene gets expressed. So actually that's something that we're very interested in, because for UPD, when you're adding a therapeutic, we don't actually know what happens there. Do you get too much reactivation or not? And so one of the things that we're interested in is, could you tune how much UBE3A and even overexpress it and control that in a very tightly regulated way, and then be able to study what happens there. So yeah, the other kind of approaches that we're putting in these landing pads that allow us to flip in different genes in that way.
Okay, that was my next question, because I didn't know what the landing pad was. And I knew you were working on a landing pad, so I was going to have you describe that.
Yeah.
So what you mean by, can you describe the landing pad again? You just got ready to start doing that.
Yeah, so I think one of the things that I talking to ASF and FAST and a lot of the investigators and families was that struck me was that Angelman is not, Angelman is very diverse. And if we want to be able to address that diversity, we need a way to quickly create many of these tools that have different genetic changes. And so landing pads are these tools that that allow you to more quickly edit the genome of cells. So conventionally making one edit to a cell line is actually very difficult, and it's very low efficiency to do that process. So what we're doing is putting in this landing pad, which is a sequence of DNA. We're putting that in first. And then the landing pad has these special elements or sequences to them that allow you to then flip things into that landing pad, other sequences, at a higher efficiency and more easily. So our hope is that there's over 20 different UBE3A mutations, for example, to create every single mutation would take a very long time by conventional means. If we put a landing pad in there first, we could then just ship that cell line to a whole bunch of investigators around the world, and everybody could do their flip a different gene or different mutation, and hopefully that would be a much more rapid process for generating these models.
Wow, cool. So you're sort of giving a blank canvas to all these different investigators, and they can all then use it how they wish. But don't you have to put the landing pad in between each spot where you would want to flip it, or no?
There's kind of two different approaches. One is more of a synthetic biology approach, meaning we want to have as tightly controlled, as least interference from the kind of endogenous, the host, how things naturally work in the cell. And so one way we can do this is put the landing pad into what we call a safe harbor site, meaning the rest of the cell doesn't really affect what's going on at that site in the genome. And then we can put whatever genes into that landing pad site, including UBE3A, and we can actually tune the level of UBE3A from that site, and just directly control it. We're not relying on anything endogenous within the cell. The other approach would be to try to put it into the endogenous, the native places in the genome. And yes, in that case, there could be a variety of different kind of arrangements, places to put it.
What's really interesting about this is that it is basic research that is directly translatable, meaning in my mind that you're finding out really important things about how human DNA works and about what UBE3A does and where it does it and when it does it, that are just really basic, and yet at the same time might be really the piece of the puzzle that we need to know to be able to get kids treated. I mean, that's pretty cool.
Yeah, I think there's a couple things on the more basic side that really excite me about how they could lead to translational applications is that one is that we don't know what a lot of these other genes might be doing, and GABA receptors are clearly going to be important, and so if we can see how those other genes affect the physiology of these cells, that could actually identify treatment options that are not directly related to UBE3A, actually. So just identifying other paths of therapeutic intervention. The other aspect is high throughput screening, like you mentioned. So one of the really nice things about models in a dish, in vitro models, so we call, is that we can do screening on them. We can put them into these plates that have hundreds or thousands of individual wells, and then screen many components. And that's actually what that's how indotecan and topotecan were initially identified, and how they screen a lot of ASOs and Cas9 approaches as well. And so actually, yeah, and then so actually in one of the projects we're working on is to create a reporter cell line that can be used for high throughput screening, using very similar kind of genome editing approaches that I described previously.
So we're already over time, and I still have a zillion more questions. So I think we're just going to have to follow up later, and maybe you'll have even more to talk about then.
Sure.
This is really interesting and exciting work, and it might be kind of hard for people to really
Uh,
understand what you're doing, but what you're doing is making a model of Angelman syndrome that you can study in a dish that's not from a mouse, but it's from actual human cells. And it's going to be probably giving us clues far into the future, maybe even after we find a treatment that helps our kids. We still might need to know how it works and how well it works and how much we need to dose it and all those other things. That this will all be something that'll be that we can find out through the work that you're doing. Yeah,
I think one of the things I'm most excited about is just hopefully we will be able to, using these systems, um, capture UPD, ICD, class 1, class 2, and more kind of genetic um, diversity, because a lot of the treatments um, that are being developed are developed for uh, loss of UB3A, but maybe with these systems we'll be able to not leave anybody behind, I guess.
that's fantastic. Well, thank you so much, Dr. Keung. It's been a pleasure to meet you for the first time, and I will hope to talk to you again soon.
Yeah, you too.
Thanks so much. Have a
Great to
great
have you.
day.
You too.